Hello, everyone. My name is Felix, and this summer I studied incompatible grafting in the Frank lab. So what is grafting? Essentially, it's like Frankenstein. Essentially, you take different parts of a plant and merge them together. So you take the root of one plant and the shoot of another to create a whole new entire plant. Now, in order for a graft to be successful, the vascular tissue between the root and the shoot must connect with each other. This is known as a compatible graft. And when this doesn't connect, this is known as an incompatible graft. Currently, there aren't really many reasons like that we know why grafting, the causes of graft incompatibility. But in our lab, we found that in incompatible grafts, there is this upregulation of genes involved in immunity-based response processes. The way an immune response works is that you have a foreign signal that binds to a receptor, and then that causes a signal cascade, which then leads to an immune-based response, such as cell death and developmental delay. Based on this, we hypothesize that immune response plays a role in graft incompatibility. We believe that there are two possible types of immune-based responses. The first response is an acceptance response, where one part of the plant will recognize the other part of the plant as itself, creating a compatible graft. Whereas in a rejection response, we have one part of the plant that recognizes the other part of the plant as a threat. And that creates an immune-based response that then leads to an incompatible graft. To test our hypothesis, we performed a mutant screen of various disease response pathway genes to identify a gene that has a significantly different phenotype compared to its mutant. We did this through a robust and efficient staining method that takes less than 1 minute per sample. And so we were able to do this by cutting a section of the plant at the graft junction, staining it using a toluidine blue dye, and then image it. And through this, we were able to identify one gene that has a significantly different phenotype known as ROS-RF. There are two possible phenotypes we could have seen from ROS-RF. Either we have a compatible graft that is now incompatible, or an incompatible graft that is now compatible. And when we looked at our results, the first thing we wanted to see was the vascular tissue, which is stained in this really pretty blue color, and then see if vascular tissue reconnects at the graft junction, which is denoted by the black arrows. When we knock out ROS-RF, we notice that there is not a difference in compatibility in incompatible grafts, as you'll notice that the vascular tissue does not reconnect at the graft site in incompatible grafts. However, in compatible grafts, we do notice that there is a difference, as our once compatible grafts are now incompatible, as the vascular tissue does not reconnect at the graft junction. This suggests that ROS-RF plays an important role in graft compatibility, and that leads us to conclude that immune-based acceptance responses contribute to grafting compatibility.
So could you tell me a little bit about the purpose of grafting?
So grafting is essentially a technique that's used to introduce different characteristics that build resistance to plants, such as cold tolerance and stress tolerance. But really, this is an easier way, because compared to modern technologies, this is so much faster and so much more efficient, because with modern technologies, it takes a really long time. So this is really just a more efficient way.
Thank you so much.
Thank you.